In this recording, we'll be going over the microscope lab. But before we begin, I want to bring your attention to some general lab logistics in this course. Under the lab section of each unit's module on campus, you will find a file titled Unit Lab Worksheet. So it'll be Unit 1 Lab Worksheet, Unit 2 Lab Worksheet, and etc. This is the document you may want to print out since it will contain close to everything you need to know for the practical. If there's any minor changes that are made, I will make sure to make you aware. But these lab worksheets will be the basis of what you need to study for the lab practicals. Next, you'll notice the first page of this worksheet is designed similar to the course syllabus, in that there is a table of contents on the first page. You can click on these topics to jump to the corresponding section in the document. And this also serves as an overview for what you can expect for on the units lab. Okay, so I can do, give me a sec, I can do that, okay? Now we're ready to begin. Page 2 of this lab worksheet is titled Anatomy Worksheet 1.0, Anatomical Position, Directional Terms, and Planes of Section. This section, or this page specifically, will not be on your practical. All of these information are already in your lecture notes for Lecture 1, which is titled Intro to Anatomy. So this information will be on your lecture exam. So don't cross it out here though, because this page is for your additional practice. Reading the lecture notes and listening to me talk are both passive learning. However, when you go through this worksheet to prepare for exam 1, now that's active learning. So I want you to do active learning in this class. Okay? So practice, practice, practice. When you have to revisit these terms during your final exam study for this course, you'll realize just how important these terms are in anatomy. We will be using these terms in pretty much every single class. And since most of you are going to healthcare field, guess what? You'll be coming across these terms on almost daily basis as a nurse as well. So I guess there's a reason that it's on the very first lecture in this course, right? So this stuff you can study for a lecture exam. It's not going to be on the practical. Moving on, page 3 here is titled Anatomy Worksheet 1.1, the Compound Microscope. For the summer 2020 course, since we won't have a chance to get you to use the microscope, I figure it's a bit meaningless to test you on proper usage of microscope. So you can ignore most of what's on this page with a few exceptions, which I'll mention in just a few seconds. But for 4 courses and beyond, if this course is able to meet on campus for labs, you will still be responsible for knowing the microscope components. Now, for the summer 2020 folks, here's what you need to know for this page. The eyepiece, which is also known as the ocular lens, these are the lenses that you look through to see the specimen. Okay. And generally speaking, this eyepiece contains, or the ocular lens has a 10-fold power magnification. That means the image you see will be 10 times bigger than they are in reality if you were only looking through these eyepiece. Now, the next thing you need to know are the objective lenses. There are objective lenses you can change in between on each microscope. Okay. And the power of magnification can range from 4 times to 100 times. These are not numbers you have to know. You don't have to memorize these 5 times, 10 times, 40 times, 63 times. You do not have to memorize those. But what I do want you to understand is that the scanning objective lens is always the lowest power lens. So it magnifies, of all the objective lenses, it magnifies to the smallest degree. The next level up is the low power objective lens. Then you have high power, and you have hydride power. Okay, some microscopes may only have the scanning power, which they also call low power. Okay, and then there's high power. Okay, And so the terminology itself, just know the order in which these would lie in. And remember the tenfold magnification on the ocular lens. That is information you'll need for the practical to do a calculation. Okay. And don't stress out too much about microscope because really there are only 2 questions of the 54. So there are 54 questions on the practical right now, and only 2 are on microscope. Okay. In a bit, I'll even tell you what those 2 questions are going to be about. Okay. Now moving on to page 4 here. This section now talks about the field of view, the concept of field of view. So what field of view is just the area that you see through the microscope. Okay, so when you look through the microscope's ocular lens, you're going to see this round circular area within which you find your specimen magnified. Okay, so the image you are looking at is actually going to be more than 10 times larger than in real life. And that's because what you're looking through are essentially more than one set of lenses. Okay, there's a series of 2 sets of lenses. Okay, the ocular and the objective. Okay, and so this page also talks about the concept of total magnification. I will post videos for you to watch, but essentially total magnification just equals to the power of the ocular lens times the power of the objective lens. So whichever objective lens you're using, if it's 40 times, okay, you will multiply that by the 10 times from the ocular lens and get 400 times. Okay, so the total magnification becomes 400 times. That means the image you see will be 400 times bigger than in real life. You don't have to, so the summer 2020 folks, you don't have to worry about these true-false questions because that's going to be on the stuff that you won't get to experience hands-on. Okay, this formula sums up the relationship between the power of the objective lens and the field size, or the size of the field of view. Okay, these two have an inversely proportional relationship. That means when you increase the power of the objective lens, that field of view will decrease. Okay, and that should make sense because if you imagine yourself zooming closer, zooming in onto something, right, you begin to see smaller and smaller part of that object. However, you're now getting a more magnified, a closer look at that specific region of the object. Okay. Now, I'll also post a video going over the calculation and the concept of field of view. Then we move on to talking about, this section talks about the famous letter E slide, and there's another slide called the depth of perception slide. And I'll post videos on those. Make sure you watch them. Okay. I do want to briefly mention, in case that it's not clear from the video that I will post. This part, number 4, goes over that the concept that when you look through a microscope, the image that you see is going to be an inverse of that image, that object in reality. Okay, so in this example here, this E, okay, it's going to be, this is how your slide is going to look like, right? The letter E on it. And then, but when you look through the microscope, somehow the E becomes inverted, or not just inverted, inversed. Okay, so essentially just take this E and rotate it 180 degrees. So you just flip the paper upside down. This is what you're going to see. On the test, there's a question that's going to ask you, which of the following is going to be how the object or that letter appears through the microscope? It could be any letter A through Z. Okay, so try practicing with a few letters. I'll give you an example. The letter B. How is that going to appear under the microscope? Well, if you rotate this 180 degrees, you're going to get something that looks like this, right? If you rotate letter J, it should look like this, okay? This part of the worksheet also goes over the concept that when you move the slide to the right, and then you look through the microscope, you'll notice the image is actually moved to the left. So it's moving in the opposite direction, but that stuff is not on the practical, for at least for the summer course. So the summer course, there are 2 questions on the practical regarding the microscope. One has you determine which of the choices that's offered is going to be the image for that specific letter through the microscope. And then the second question on microscope is going to have you go over this calculation for field of view size. Okay, and I'll post a video on that. Please let me know if you have questions. You don't have to worry about number 7 that goes over estimating the size of the red blood cells. Okay, not for the summer folks. And that concludes our video for the microscope lab. So remember to watch the videos that I will post.